I'm a graduate student in the Prober Lab where we use zebrafish larvae to study sleep. I think sleep is one of the most interesting biological processes because we all sleep every night, give or take, and yet there's still so much that we don't know about it. So my thesis focuses on how one particular molecule, melatonin, regulates sleep. You've all probably heard of melatonin. It's available over the counter as a sleep aid, and most people have some first or secondhand story about its effectiveness. In fact, since its discovery, there have been a number of conflicting reports that have led to a lot of doubts and controversies about whether or not melatonin is actually sleep promoting. We make melatonins in our own brain in a structure called the pineal gland, and its synthesis is in rhythm with the 24-hour clock, with a peak of melatonin production at night. Now, if you want to know if a molecule is important for a process, the best thing to do is remove that molecule and then ask if that process is effective. So the Prober Lab removed melatonin from zebrafish by deleting the gene responsible for its synthesis. These fish don't make melatonin, and they slept about half as much as normal fish. So this suggests that melatonin made in the zebrafish's own brain is essential for nightly sleep. Now, I wanted to follow up on this and use zebrafish to understand how melatonin is promoting sleep, and I tried to do this in a couple of ways. First, molecules like melatonin usually work by binding a receptor, and there are two melatonin receptors. They're called MT1 and MT2, and it's totally unknown in any animal if one or both of these receptors is required for sleep. So to figure this out, like I said before, I need to remove these receptors and then look for changes in sleep. So I found that MT1 mutants were strongly impaired for sleep, just like animals that don't make melatonin, and I did not see this effect in the MT2 mutants, because they slept normally. Additionally, when I give melatonin, MT1 mutants they failed to fall asleep, that is, they were resistant to melatonin. So these experiments suggest that melatonin is acting through the MT1 receptor to promote sleep and not the MT2 receptor. Now that I know what the relevant receptor is, I wanted to know where in the brain melatonin is acting, and this will give us a clue as to how it promotes sleep. I found that melatonin receptors were expressed in brain areas that also have to do with visual processing. So this led me to hypothesize that maybe melatonin promotes sleep, at least in part, by turning down our brain's responses to light and visual input. When we sleep at night, we have what's called an increased arousal threshold. This means that we're less likely to respond to stimuli that normally we would respond to when we're awake. This is a hallmark of sleep, but it's not really known how the brain does this. I think melatonin is playing a key role in this, which is really exciting, and I'm currently developing assays to test this hypothesis. Through my findings, I hope to shed new light on how this popular yet controversial molecule regulates sleep. Thank you.